Hola, buenos días, buenas tardes, perdón, estimados invitados, docentes, alumnos, personal administrativo que nos acompaña este día. Sean bienvenidos a la presentación del Seminario Departamental del Laboratorio de Biotecnología Genómica, que será presentado por el doctor Abulude Ibukun John. Dice, alumno egresado del doctorado en Ciencias en Biotecnología, adscrito al Laboratorio de Biotecnología Genómica, estancia doctoral realizada en Tufts University School of Medicine, Boston, United States. De maestría realizada en Obafemi Awolowo University, en Nigeria. El trabajo que va a presentar está relacionado con la línea del mecanismo de las interacciones bacterianas, específicamente en bacterias depredadoras, base para la caracterización fenotípica y molecular de las investigaciones de este tipo de bacterias. Adelante, doctor.
Hello. Good afternoon, everyone. I'm Abulude John, and I'm presenting the novel protein-protein interaction among predation essential hypothetical proteins in Bdellovibrio Bacteriovorus attack phase, discovered using AI, specifically AlphaFold-Multimer. So this work is part of my thesis, my doctoral thesis here, and was directed by Dr. Jean Wu and Dr. Daniel Kadari of Rutgers School of Medicine. So why do we even care about looking at Bdellovibrio? Why do we want to study Bdellovibrio? We all know that there's a rising case of resistance to pathogens, which is a big concern now because a lot of bacteria that are pathogenic are resisting the traditional antibiotics that we all use, and we need alternative means of control of these bugs. So Bdellovibrio Bacteriovorus, which is a predator, has turned out to be a very potential candidate in the combating of multi-drug resistance organism, and that's why we really care about studying Bdellovibrio. So I'll introduce Bdellovibrio to us. Bdellovibrio is a very small gram-negative bacteria of about 0.25 to 0.4 micrometers, and we can see the diagram of Bdellovibrio over here. They are obligate predatory bacteria. Excuse me, So they are obligate predatory bacteria that prey on gram-negative bacteria for nutrients. So they cannot survive really without the host. Although some forms have been isolated independent of the host. The predation strategy among Bdellovibrio include epibiotic and intraperiplasmic predation. So in epibiotic predation, the predator attaches to the prey just like this, and it does not enter the prey. It just consumes the prey from outside, and that's why we call it epibiotic. But in interperiplasmic predation, the predator attaches to the prey, but as well it enters into the prey and establishes itself in the prey and destroys the prey from within. Interperiplasmic attach and enter and consume their prey from within, just like I said. So here is a life cycle of the interperiplasmic Bdellovibrio. We are focusing on the interperiplasmic Bdellovibrio. So the attack phase consists of Bdellovibrio moving very fast and attaching itself with the prey. Then it enters into the prey, and because we're talking about interperiplasmic, it goes and establishes itself in the periplasm of the prey. So from there, it starts growing by using the prey nutrients, the prey contents. Then when the prey content or the nutrient is depleted, the Bdellovibrio septates, it divides, and lyses the remains of the cell of the prey and escapes as free-swimming attack phase Bdellovibrio, which can continue the cycle all over again. And like I said, Bdellovibrio can also be isolated using antibiotics, selection pressure, and diaminopylamic acid to isolate them as host independent. Here are some diagrams, microscopic diagrams. Here we see the invasion process where Bdellovibrio attempts to enter the prey. And here we see its growth within the prey. So, the life cycle of Bdellovibrio has been well established. But the challenge is that many molecular aspects of Bdellovibrio predation and growth are not well understood. And these have been documented by several studies. So, here is Bdellovibrio. There are some mechanisms that allow it to be able to encounter the prey, to be able to enter the prey, to be able to penetrate the prey. A lot of them are not well understood. Its growth within the prey. Specifically, there was a report that Bdellovibrio is sensitive to a particular cue in the host, in the prey, that makes it to divide. And this cue is unknown as of now that we speak. So many molecular aspects of these organisms, the predation of this organism is not well known. So we attribute we are thinking, or we thought rather, that this unknown might just be as a direct result of a lot of hypothetical proteins that are present in Bdellovibrio. So these hypothetical proteins are proteins that have been annotated, but their functions are not known. They do not resemble anything that, we can find in other organisms like E. coli and all the other organisms. So they are unknown, and there are a lot in Bdellovibrio. So our the gaps in our knowledge might be as a result of this hypothetical protein. So what we did was try to understand the functions of these hypothetical proteins, try to start probing these hypothetical proteins so that we can understand the molecular mechanism underlying predation. So here I give a few examples. Bdellovibrio has about 30% of its genome as hypothetical proteins. That's about 1,047. In a study in 2010 that identified some genes, it identified 240 genes in Bdellovibrio which are important or which are expressed at the phase when Bdellovibrio was specifically entering the prey cells. And surprisingly, out of these 240 genes, 174 of these genes, that's about 72%, are hypothetical proteins. So it's the argument for our reason for the lack of understanding of the molecular mechanisms, is justified. Another study identified and confirmed 104 genes as essential by transposon mutagenesis. So what they did in that study, they did transposon mutagenesis, they disrupted some genes, and they found that Bdellovibrio was not able to predate. And they found 104 genes, and out of those 104, 66% constituted the largest category, and they were hypothetical proteins. So, in order to be able to explore or find, functional relations in these hypothetical proteins, we resorted to using AI, specifically AlphaFold-Multimer. So I'll talk a little about AlphaFold. So in 2021, there was a paper published, and there was a conference, which is the CASP 14, where models of protein predictions and protein foldings were tested. And what was found was that AlphaFold performed very, very well, and it demonstrated near experimental accuracy in protein structure prediction. So it has since then revolutionized the way we approach molecular analysis of protein folding in silico. So, revolutionizing protein-protein interaction, AlphaFold has improved the prediction of protein-protein interactions, enabling researchers to better understand molecular mechanisms and interactions critical for biological functions. And it has also advanced discovery of protein complexes. And now we have AlphaFold-3. As at the time of my study, we were on AlphaFold-2. But now there's AlphaFold-3. So another thought very important for the work was that protein function in conjunction with other proteins. They are a team. They don't function alone. Most proteins function together. Let's think about the flagellum, for example. It's like a machinery for movement, and it consists of a lot of proteins functioning together. So, dealing with hypothetical proteins, which are a lot in Bdellovibrio, we thought, okay, let's approach it from the angle of protein-protein interaction. So that way, we can see what proteins are functioning together, what proteins are interacting together, and that way we can begin to solve the mystery of the molecular basis of predation. So AlphaFold-Multimer was central to our to the work. So what we did as a methodology was recover data, already published data, for RNA sequencing in Bdellovibrio. So from this RNA sequencing, we focused on the proteins that were expressed in the attack phase, and we combined that with genome-wide transposon sequencing. Those are the proteins that have been discovered by transposon sequencing to be important for the predation. So we used AutoVen2 to match this data so that we can have overlaps, and from there, the overlapping proteins from the data, from our input data, we passed into strings and used that to predict protein-protein interaction by association. So we validated strings prediction because strings can predict from different, using different criteria, including searching articles and journals to see what proteins are mentioned together. So it's not really a direct interaction. So to predict a more direct interaction, we used AlphaFold-Multimer. Then our outputs from AlphaFold-Multimer showing interactions, we further analyzed with different web servers, Interpro, Microbes, Online, and all these web servers. Then we did experimental confirmation of these protein-protein interaction using the bacteria adenylase cyclase 2 hybrid system, and we had experimental positive or negative results. If the interaction was positive, then it showed a blue color on XGAL IPTM plates supplemented with antibiotics. So we show our results here. So when we gathered the data, the expression data, we had 2 of them available from publications. So we gathered attack phase protein, like I said, 1,456 attack phase protein from Lambert et al. And 411 attack phase proteins from Karonka et al. And we overlapped that with the transposon data from Duncan et al. And that was 101 proteins. So what we found was this central overlap of 33 clusters. AutoVen does a clustering. So these 33 clusters contains 43 proteins. So these 43 proteins we termed as the attack phase predation essential protein, and APPEP for short. So what we did with these 43 proteins, so what these 43 proteins represent is proteins that are expressed in the attack phase from the air and also relevant for Bdellovibrio predation. So what we did with these attack phase proteins was to make eventually a global network of protein interaction, which consisted of 169 proteins. So here shows a summary of the input proteins and the protein clusters, which are the sum of all these circles right here, and the singletons, that is the proteins that did not cluster at all in this orthovane cluster. So we took these 43 proteins, and we used each of those 43 proteins as input query for strings. And so we had 43 local networks, protein interaction networks. So here is an example of a local interaction network for the BD0739. And so in 43 places for other proteins. So we had a robust global protein interaction networks, which does not just consist of any protein. It consists of proteins that are relevant to the attack phase and that have been shown by publications or by experiments to be important for protein, for Bdellovibrio predation, excuse me. So what we then did was to cluster these proteins. There were 169 proteins. We wanted to cluster them in order to have relevant clustering for complexes and for protein-protein interaction. So we use MCL clustering in strings, and we found 27 protein clusters. So here we're showing 3 of those clusters, which are representatives and which were the ones that we further worked on. So these clusters are interesting. This cluster 10 and cluster 11, the proteins in cluster 10 form an operon. And the proteins in cluster 12 as well form an operon. The proteins in cluster 4 were mostly comprising of proteins with TPR domains and proteins with FHA domains. So here is a table showing the string combined scores for all the interactions that we analyzed, and also showing the AlphaFold prediction, AlphaFold IPTM score. So I'll explain this a bit. Here, I said earlier on that we had this global network and we found 27 clusters. So we checked the score, the string score for each of these lines, each of these network. We checked the string score and we validated those string scores using AlphaFold as well. So what we have here is the string combined score for each of the possible interactions, and what we have here is the IPTM score for each of the possible interactions. So the positive interactions were interactions with IPTM score of greater than 0.6. So the IPTM is an index in the AlphaFold that we use to measure, it's an interface PTM score that we use to measure how well, how confident the prediction is. And the IPTM score that is less than 0.5 is a random interaction, but more than 0.5 is fairly confident, but we decided to go a little above that to use 0.6. So any interaction above 0.6 is a positive protein-protein interactions predicted by AlphaFold-Multimer. So here we found that in cluster 4, we had 2 positive interactions here in cluster 4. We checked a lot, and a lot of them were negative, but we found 2 positives. And in cluster 12 and cluster 10, cluster 10 and cluster 12, which are shown here, we also found positive interaction. But the interesting thing about cluster 12 and cluster 10 was that they were in an operon, like I said, and they also formed a complex, which we were not expecting at all. We found that in AlphaFold, we were able to form complexes with these, and those complexes, the resulting IPTM score for those complexes were very high, and that's very highly confident predictions, 0.852 and 0.34. So for a bit, we focus on, I want to focus on and talk about this cluster 12 and cluster 10. Okay, but before then, we also included positive controls here. So these positive controls that we included in AlphaFold were known interactions, known experimental interactions. And these non-experimental interactions are and housekeeping proteins as well, interactions among housekeeping proteins which are established. So they also had high IPTM scores, 0.7, 0.89, 0.5. So we did a statistics to compare the IPTM scores of the predicted interaction and in our predicted interactions and positive control. So in yellow here is our known interactions, which are the positive controls, and in teal green here are the predicted interactions that we found from the clusters. So we did a mean comparison of the IPTM score, and we found that the IPTM score of both were not statistically different. So there was no significant difference in p-values of 0.19 between the two. So I talked about the two operons which were from the two clusters, clusters 12 and cluster 10. Now, the interesting thing about this operon was that when we checked the genomic structure of this operon, we found that they were very, very similar. And like we can see, the 2212 and the 2217, which are like the corresponding pairs from the two operons, were the biggest. And it maintained the structure across the operon. These were the smallest and they corresponded. So we initially thought that maybe it was just an event of duplication. But when we checked the NCBI blast percentage, we found that they were not, they were totally different percentage identities. And so corresponding pairs, 2212, 226 were different. 27, 23, 2209, which are corresponding pairs here, as they have a low percentage identity. Likewise, 27, 24, and 27, 2210, which are these, also have low. So across board, we found that they are actually different operons and could be performing different functions within the Bdellovibrio. So in AlphaFold, we generated the complex. So this was interesting because we added each of the other members of the operon to the largest protein in the bulk. So for this operon, 2212 is the largest protein. So we found that every other protein was able to interact with 2212 with a very high IPTM score, here 0.8, 0.813, and the final interaction for all the proteins coming together, 0.85. And here shows the prediction align error for AlphaFold. So I will explain this with another diagram as we move on. But maybe I can try, but I think it will be clearer with a less, with a protein interaction that is not up to 4. But let me just explain this, what this means. So this square here is the 2212, and as we can see, the biggest it is the biggest square because it's the biggest protein. Now, all the proteins are aligned along this diagonal. So here, this square right here is the 2212. And this square, is the 2210. And this square is the 2212. And this square is the 2209. So what we see here between the 2212 and the 2210 is that in this region, there is interaction. And between this and this, in this region, there is interaction. And between this and this, in this region, there is interaction. But I believe it will be clearer. I believe I can explain it clearer with interactions that have less members. Okay, so the next, in the next cluster, which was formed, which was cluster 10, and had similar, was formed by a similar operon with similar genomic structure, we can see that the very, very similar protein complex in this case, we can see, for example, We can see this pocket right here that was for the smallest protein, 22.7, and we can see the same pocket right here for the smallest protein, 22.09. So these structures are not only similar in the genomic landscape when we look at them, according to the operon, but when the protein complex, that was modeled also was very, very similar. So I now move on to discuss the third cluster, the cluster 4, which is this cluster right here. So in the cluster 4, we find, I'm sorry about this. In the cluster 4, we found BD0075 interacting with 474, and we also found the same BD0075 having positive interactions with 3743. So what I'm trying to explain here is that here is BD0075. Here is, here is BD0075, and it's able to interact with 474, and also able to interact with 3743. Now, in AlphaFold, when we tested all these other interactions, this is a string predicted interaction. When we tested all these other interactions, there was no positive interactions for AlphaFold. So in that way, we were able to eliminate the choices, the multiple choices that we have here, to focus on the most relevant one as predicted by AlphaFold, so which are the 474 and the 3743. So we did an analysis of the 474 and the 0075. So here is an AlphaFold model of the protein folding. So what we found was that the 0075 folded into 3 distinct domains, which are the A domains, or what we call the A fold, or the fold B, and the fold B and the fold C. And they are between these amino acids, 1 to 83, forming this small fold, then the fold B between these amino acids. And the same we did for 474 right here. So it also had 3 distinct folds, the fold A, and the fold B, and the fold C. So, but what we found was that all the folds had unique domains that they contained, functional domains. For example, here, the fold A of the 474 had the FHA domain, and the fold A of the fold B of the 74 had the FHA domain. The fold C had TPR domains. The same here, the fold C has TPR domain, and the fold B here has TPR domain. But the fold A does not contain any known domain. So here is also a representation of our analysis from Interpro. And what this shows of the 0075 is the region where the domains are located. So here is where the domain A is located, and it's in the cytoplasm. So what we now did was to do a multiple sequence alignment to see the conserved regions. So these are traditional multiple sequence alignments to see the conserved regions. And what we found that the domain A, this is the domain A, these are all genomes of Bdellovibrio, but specifically the BD0075 protein aligned across the genomes. So we found that the domain A is conserved, but we do not know, it has no known functional domain. So we did another analysis to show the conservation of this domain A. So in this analysis is the analysis of a conservation scores across the BD005 sequence alignment. So the green colored region in our conservation plot represents positions we score below the threshold and are considered to be statistically significant for conservation. So here is a threshold of 0.1 that we set. And any threshold that falls below this line any conservation score that falls below this line is deemed to be significantly important or significant for conservation. And here we can see that this part, just like we showed in the traditional multiple sequence alignment, is conserved. So this A part is conserved, and that's the part that corresponds to this region. So functionally, this part is important because conservation can show importance. And just like this part, other parts which contains non-functional domains, the TPR domains, are as well conserved. So since we saw conservation in this part, we wanted to know what, we wanted to check out or probe further to see what can we find in terms of resemblance to this domain. So we did a structural homology search with Swiss model, and we found an interesting result. We found that this domain corresponded to the GYF domain in humans, which is the CD2 binding protein domain, which is shown in red. So we see this GYF domain, and in blue, we see the 0075 protein, and this portion of the blue is the A fold. And the GYF domain, when we overlap it structurally with the A domain, we find this structural resemblance. So this GYF domain of the human CD2 binding protein structurally resembles the A fold of BD0075. And so we did not leave it at that. We went to NCBI CDD to check to align the known or the representative sequences for the GYF domain with our Bdellovibrio Bacteriovorus. So here we can see the representative GYF domain from chicken, from yeast, from humans, and we can see the GYF domain from Bdellovibrio Bacteriovorus, and we can see this conserved region right here, which is GYF. Okay, so like that, we're able to decipher functionally what this might resemble and what it could be doing functionally in Bdellovibrio. So the interaction, so I already discussed the structure of the 0075 and the structure of the 474. So we look now at the interaction between 0075 and 474. So in AlphaFold, scores that are in blue in these interactions are greater than 90, and they are shown in blue and are very high confidence. So we can see here the interaction between, in the purple we have 0075, and in the green we have 474. And so maybe I can explain a little clearer with these predicted align plots. So what we see in this square right here is 0075, and this other square right here is 474. So if there is interaction between this square and this square, we're going to know from the green shade in these other two squares right here. So that's how we read the predicted align error plot. So, and as we can see, here is in this 0075, we can see the A domain right here, we can see the B domain, and we can see the C domain. And here in the 474, we can see the A domain, the B domain, and the C domain. And here we can see that this region of interaction corresponds with the C domain of 474 and also the C domain of 0075. So the C domain is where the interaction is predicted to occur, to happen. And here I'm just showing one of the representatives positive controls that we also used. So here is one of the proteins, and here is the second protein having 2 domains. Okay, so we found another interesting thing, and that was that BD075 interacts with 474, just like I showed here, but not only does BD075 interact with 474, BD075 also interacts with 3743, an hypothetical protein as well. But the interesting thing is that they interact at exactly the same region. So this table shows the amino acid residues that are interacting in BD075, and here are the amino acid residues interacting in 474. Here are the amino acid residues interacting in 3743. And what we could see was that these amino acid residues, which I have colored in blue, are the same amino acid residues responsible for the interaction in 474 with 75 and in 3743 with 75. And that was an interesting thing that we found. So we thought that this could be some kind of competition or competitive binding for the same site amongst these 2 proteins. Now, if we look at the IPTM score of 75 and 474, we found that it is 0.619 and 0.676. So this has a higher IPTM score, and we thought, oh, surely maybe it must then have a preference for this 3743 and maybe less preference for the interaction with 4743. So we decided to simulate interaction dynamics using the 3 proteins at the same time to see what the preference was. So here we could see that, just like I showed earlier on, the interaction between 75 and 474, and here is the interaction between 75 and 3743. And we could also see that that interaction occurs. But when we put the 3 together, we find that it no longer binds to 3743, showing a preferential binding for 474 in the presence of 4743. So this was interesting to us, and when we look, which I'm going to talk about a little, but I can give a hint right here. So, we found that here, these 3 proteins, now we were seeing this kind of competition between them. We're now interested in where exactly, at what time point or at what phase were these proteins expressed. So we found that 75 is expressed in the attack phase, 474 is expressed in the attack phase, and 3743 is expressed in both phases, both in the attack phase and in the growth phase. And what that could imply is that the preference for 474 in the presence of 37, which is also present in the attack phase, but in the attack phase, all these 3 are present, but only these 2 are binding. 474 is preferentially interacting with 474 is preferentially interacting with 75. So, functionally, this could mean that this interaction is taking place, and when the levels of 474 drops, then 75 is able to interact with 3743. And then we checked a co-expression of, we wanted to see the co-expression partners of 75. So we found that 474, just like predicted, is a co-expression partner with a Pearson correlation of 0.99, and which means that they are co-expressed at the same time points, just like you can see from the overlay gene expression profile right here, that at the same time point when 474, this 75, at the same time point when 75 is decreasing, 474 is also decreasing. At the same time point when 75 is increasing, 474 is also increasing. And so the overlaying gene expression shows us that they are available for interaction in the cell, in Bdellovibrio at the same time. So here we show from our findings, from what we've discussed earlier, we show a cartoon interaction between 75 and 474, showing the B and C domains interacting, the C domains of the two proteins interacting, and the GY domain that we found. So we wanted to confirm this prediction with experiments. So we used the two hybrid system for experimental confirmation of the interaction. So what we did here was to clone one of the proteins into this plasmid containing a kanamycin resistant. So this is the T20. This plasmid contains the T25 fragment of adenylate cyclase enzyme, and the other plasmid also contains the T28 fragment of the adenylate cyclase enzyme. So the method is to clone each of the interacting proteins into each one of these plasmids. And by so doing, it brings, if these two proteins are indeed interacting, X and Y comes together and brings the T25 and the T28 fragments together, and that will reconstitute adenylate cyclase in the cell. And that can be seen visibly by the breakdown of S-gal on the plates to give off blue color. So blue colonies will represent positive interactions. So when we did the two hybrid assays, these are positive control, which is the 18 fragment and the T25 fragment coming together using a leucine zipper, which brings them together. And we see that's positive by the blue color. Then we add the empty plasmids, which were just the T25 and the T18 fragments without the leucine zippers, which are not expected to come together. So that was negative. Then we see here, like, we did not only test the positive interactions that we found, we also test a number of negative interactions. This 76, which is a close neighbor of the 75, was negative. The interaction did not interact with 75, and 75 did not interact with 472, which is in the same operon with 474. And here is 75 and 474, showing a positive interaction, which is a confirmation of the interaction that we predicted. So, also, we show that 75 does not interact with 471, and we also show, this is another time point where we repeated the experiment, also showing that the 75 interacts with 474. And here we show that the 474 does not interact with the 472. Now, we also wanted to see, from our predictions using AlphaFold, we said that the C domains of the 0075 and the C domains of the 474 were the ones that were in interaction. So what we did was to delete domains from the 75 and from the 474. So we deleted, if I can go back a little to this picture. Here we can see the complete 75. Oh, excuse me, no worry. I can use this. Here we see the complete 75 and we see the complete 474. And we see that there is an interaction shown by this positive blue color. Now, when we remove or when we deleted all the domains of 75, just leaving the A domain, which is in the cytoplasm, predicted to be in the cytoplasm, we found that there was no interaction. Now, we included in another protein species, we included the transmembrane domain, because these are transmembrane domain, like I said earlier on. So this, we included the transmembrane domain with the 75, the A domain of the 75, and we found a slight interaction showing that the transmembrane might be involved in the stabilization of the complex. And this is a phenomenon that has been reported, severally in different works, that the transmembrane domain can indeed be a part of the protein interaction and for stabilization of the protein complex within the membrane. And then when we added in another protein engineering, when we added the C domain to the A domain, we got back the interaction, showing that indeed, just like we predicted, the C domain is involved in this interaction. So we did something similar also for the 474 protein. So for the 474 protein, this is the like a flip of what we did for the 75. So for the 474 protein, we left only the A and the B domains, which were in the cytoplasm, without the C domain and the transmembrane, and that was negative. Then the 474A domain alone, with the C domain, was also able to restore the interaction. And so we confirmed that indeed the C domain was was important for this interaction. So we also calculated physicochemical properties for the proteins, all the proteins that we studied. So the interesting thing here is that we found that 474 is an unstable protein. So a stability index that is greater than 40 signify an unstable protein, and the instability index of 474 was calculated at 44.86, which shows that it's an unstable protein. And now it makes a lot of sense, because if we remember, I said that 474 is available for interaction, preferential interaction, may be available for preferential interaction with 75 at the attack phase. And so when it depletes in the cell, there is an opportunity for 75 to also interact with that 743. Also, we calculated the binding affinities for the protein complexes and the protein interactions that we studied. So a negative delta G binding affinity shows that the protein complex readily can come together. And these are high values that was calculated for 75 and 474 at -19. So this shows that this interaction is very, very possible, and which requires less energy of formation. And as well, now the interesting thing was that, just as was shown with the preferential binding of our interaction model in AlphaFold, we've also found with this result that the delta G of 75 interacting with 37 was lower to the 75 interacting with 474. So this can easily form, and it corroborates the results from AlphaFold, that this interaction can be formed easily than this, and that this interaction will happen in the presence of 3743, which is present in both phases of the Bdellovibrio predation. So, and other ones that we show here, the other, for the complexes, we found 67.7 and 50.6, which are very high binding affinities. So another thing that we did was to mutate specific amino acids. So we wanted to see the effects of specific amino acids in the interaction of 474 with 475. So we already said that the interaction is occurring at the C domain, and that's so in the C domain, the residues that we showed earlier on, some of them are salt bridges, which are the strongest type of protein-protein interaction that we could have. So we targeted some of those. So these are the residues that are involved in the interaction between 74 and 75. So the ones that are highlighted are the salt bridges, which are the strongest interactions holding this complex together at a distance of 3.5 Armstrong. So what we did was to target specifically 0 arginine 806 and also lysine 765. So we targeted these two, and we did mutation analysis for them. So this the technique we used in making the mutation is called splicing by extension overlap PCR. So in which we had 4 primers, primer 1, F1 and F2, and F1 and R1, and F2 and R2. So what we did was to introduce the mutations into the inner primers, which are the F2 and R1. And so we did a SOE PCR, as it is called, to get the mutations. So this just shows a rundown of the methodology of the cloning after the mutation and everything. So the SOE PCR products, when we got them, we purified them, and we digested them with restriction enzymes, purified them with magnetic beads. Then on the other side, we digested the respective plasmid, which I spoke about earlier, purified them also, and did a ligation in a standard cloning procedure, and transformed those into E. coli X1 blue, extracted the plasmids, and interacted the mutant species of 075, which was generated by this process, with 474. So this also shows a process of the methodology. So what we did was to create single mutations and also double mutations. So we targeted 75 alone, and we targeted 806 alone. And as well, we targeted 75 and 806 in a double mutation. So this shows some gel electrophoresis from the process. And we sent the resulting protein, the resulting amino acid, the protein for sequencing. And when it came back, we found that the only point where the there was changes in the nucleotide was at the point where we introduced mutation for 765, and the same for 806. The only changes were in the points where we introduced the mutation. So we sequenced to confirm that the mutation that was done, the point mutation that was done was accurate. So then we interacted this protein species with the 474. And what we found, this is the Y-type interactions, which I had spoken about earlier, the 0075 and the 474 in the positive interaction. Then the single mutation also showed a positive interaction, showing that this interaction could not be broken by a single mutation at 765, and also by single mutation at 806, the interaction could not be broken. Then by double mutation as well, we found that the interaction still remained intact. So this shows a strong interaction between the 075 and the 474. And these are the positive controls. This is an interaction overview of the mutation experiment that we did. So I move on to the discussion. So what we were able to do was create a robust global interaction network of proteins in protein interaction for essential attack phase protein involving 169 proteins. From this, we explored 3 out of 27. So we have other protein interactions, relevant functional protein interactions that are yet unexplored. But from these 3 that we explored, we were able to use cutting-edge tools and experiments to, beyond the doubt, confirm the interaction between 75 and 474. 75 exhibits a stable interaction with 474, and this interaction could not be compromised by single mutations converting lysine to to D at this point. Furthermore, double mutation in these positions did not hinder the interaction. The stability of the interaction may be due to multiple bonding that exist at the contact surface of the two proteins. So we also establish that 75 and 47C domains are involved in the interaction. An analysis of physicochemical properties shows that 474 is unstable, and it can benefit from its association with 075 and may be important for its predatory role. Our model of this 75, 474, and 73, 43 shows that the interaction dynamics shows the ability of 075 to preferentially interact with 474 in the presence of 3743 in the attack phase, and may allow the interaction of 3743 to bind at the growth phase when the cellular level of 474 drops. We also have identified two novel complexes formed by two operon with similar genetic arrangement and have been discovered with confidence interaction of IPTM of 0.852, which is a very high and matches the stringent cutoff used of IPTM 0.585 used by O'Reilly to describe the most confident complexes. The second, the BD2726 complex has a high score as well of 0.37. Although these complexes seem structurally similar, blast identity shows them that they are different, and they might suggesting that they are functionally different as well. So in conclusion, using two RNA sequencing data and a genome-wide transposon sequencing data, this study provides a robust functional interaction network comprising of 169 proteins clustered into 23 clusters, which was generated from 43 predation essential attack phase protein. We've explored 3 of these 23, and with our approach using AlphaFold, we found novel protein-protein interactions and complexes demonstrating the potential for further discovery among the remaining clusters yet to be explored. We validated 75 with 474 interacting and showed that, as predicted by AlphaFold, 75, the C folds of both proteins are involved in the interaction. Furthermore, this study is the first to report the protein complexes involve two paralogous operon, the operon 2209 to 2212 and the operon 2723 to 2726, which are both expressed in the attack phase. And further work will focus on experimental exploration of these novel interactions and complexes. So, taken together, our approach has not only discovered functional novel interactions and complexes, but has also provided templates and resources for further discovery and exploration of this protein interaction with an implication for the understanding of the molecular mechanism of Bdellovibrio Bacteriovorus prediction. So, I acknowledge the government of Mexico for allowing me to come to Mexico to study. I acknowledge the Instituto Politécnico Nacional, our great school, for providing a conducive environment for me. I acknowledge CONACYT for the scholarship that was awarded me. I acknowledge BEFI for the regular scholarships that is being given to support us. I acknowledge the DRI for the opportunity to have my estancia in the United States. And I acknowledge my thesis directors, I acknowledge the members of my committee, and I acknowledge Dr. Andrew Kameli, with whom I stayed in Boston, Tufts University, and my mentor from Nigeria, my family, and all my lab members. Thank you.
Bueno, pues buenas tardes. No sé si pasaremos a la sesión de preguntas y respuestas. ¿Alguno de los alumnos que tenga alguna pregunta?
Congratulations by your works. Congratulations on your works.
Thank you.
It seems to me you have done a really interesting job. So I have a question. Why are you using genome-wide transposon sequencing data?
Okay, so the genome-wide transposon sequencing data reveals to us the genes, which by implication, the proteins that are expressed in Bdellovibrio, because not all genes will be expressed, as we all know. Some genes are not expressed, though they are genes. They are not expressed as proteins. You know, they don't go to proteins. They just stop at the level of RNA. And so, specifically, our work focused on the attack phase protein. So we wanted to see what proteins or what genes were relevant, you know, to Bdellovibrio predation at the attack phase. So we gleaned data from genome-wide transposon, from genome-wide RNA sequencing data to be able to find the attack phase proteins that were expressed, because we need them to be expressed and we need them to be relevant. So that was why we combined the genome-wide RNA-seq data and the genome-wide transposon sequencing data. I hope I answered your question.
Okay. I have another question.
Yeah.
So, how can you do another validation, experimental validation of your results?
What? What did you say? I didn't hear. Sorry. I didn't get the question.
Okay, I'll repeat it. How can you do another experimental validation of your results?
Oh yeah. Okay. So the experimental validation of our results we did by the Bacteria-2 hybrid assay, which I already showed. So all these are experimental validations of the interaction between 0075 and 474. So further work can be done on the validation of the complexes. Now, the complexes which were found, which were the 2212 complex and the 2726 complex, we can use other methods, you know, experimentally to validate by pull-down assay and those kind of methods we can use to validate those. So further work can definitely be done. And don't let's forget also that we've only explored 3 clusters, you know, and found this much. So we have 24 clusters, you know, that contains a lot of hypothetical proteins, you know, that can further be experimentally explored as well, you know, working or stepping to understanding the mechanism of Bdellovibrio predation.
Thank you.
Yeah, you're welcome.
Hello.
Hello.
Thank you for, congratulations for your presentation. More than a question, it's a curiosity of the terminology you used. You said the proteins, those that were related, just in the end of the presentation, you are still calling them hypothetical proteins. What should we need to be done to call them? I mean, you already find some connection between them. Why are you still calling them hypothetical protein?
Okay. Well, now, thank you for the question. It's a very good question. So, the work to unravel the hypothetical proteins is not just going to be something that we can do overnight, you know. So, what we've been able to establish here is a link. Now, we should remember that when we talk about hypothetical proteins, they are proteins that the functions are unknown, one, and they are not compared, you know, they can't be compared to any other protein that we see in other organisms. So, the function, we've been able to establish a molecular function 005 by prediction and by experimental validation. But we still need to find out by other means, experimental means, in Bdellovibrio itself. Because bacterial 2 hybrid system is an E. coli-based system, not a Bdellovibrio-based system. So we need to translate these results as well into Bdellovibrio. If I'm making sense. Okay, then we can associate this to specific functions. Now we are dealing at the level of the molecular mechanism, which was intuitive for us to do because these are unknowns. So the first point of call for trying to unravel this mystery is proteins don't work alone. So we want to see what are they interacting with. I think of it as like a simultaneous equation. For example, if we have one equation that contains 2 unknowns, we might not be able to solve it. But when we have the second equation, then it becomes a simultaneous equation that can be, solved by substitution. I hope you get me. Thank you.
Thank you.
You're welcome.
¿Alguien más que tenga alguna pregunta? Procederemos a entregar un reconocimiento. Voy a leerlo muy brevemente. Dice el Instituto Politécnico Nacional, Centro de Biotecnología Genómica, otorga el presente reconocimiento a Ibukun Abulude por su destacada participación como ponente en el Seminario Departamental del Laboratorio de Biotecnología Genómica. Ciudad Reynosa, Tamaulipas, 28 de octubre del 2024. Atentamente, la técnica al servicio de la patria, lo firma nuestra directora, doctora Amanda Alejandra Oliver Hernández. Doctor. Bueno, pues muchas gracias, compañeros, por su participación.